This is Lecture 4, Part C, Measuring Microbial Growth. So population growth is how we measure the growth of microbes, and this is accomplished in a couple of ways. First, we can track changes in the number of cells by directly counting the cells, or second, we can assess changes in population by assessing the level of some cellular component such as protein, nucleic acids, or dry weight. Those are all considered indirect measures of microbial growth. So direct measurements involve directly counting cells, or evidence that cells were present. This can include microscopic counts that are derived using routine light microscopy, including phase contrast or bright field, or it can also involve microscopic counts using fluorescence microscopy. Another method that is considered to be a direct measurement of populations is called the use of the flow cytometer. Alright, let's talk about microscopic counts, and let's talk about a very traditional way to do that, which is called the Petrov-Hauser chamber. So to use a Petrov-Hauser chamber, you need to begin with a sufficiently diluted sample so that cells will be spread apart when you look at them. You then simply pipette the sample into the chamber, which is designed to have a grid. The grid is shown here, and a zoom out of the grid, or a portion of the grid, is shown here. So you simply pipette the sample into the chamber, and this is designed to have a grid that depicts an area where a tiny well has been cut, and the well is of known volume. Typically, that known volume is 0.02 millimeter cubed. That's what you'll see in a standard Petrov-Hauser chamber. If it's different, it'll be written on the Petrov-Hauser chamber itself. The grid is divided up into areas where there is a double line bordering what are considered the large squares. So this is what I mean by the double line right here. That's the double line. Okay. Each of those large scale squares holds 16 smaller squares. And in total, there are 25 of those large double-bordered squares in the Petrov-Hauser chamber, or covering the Petrov-Hauser chamber. So what you do is you count several of those large double-bordered squares and take the average of that count. You then relate this number of cells to a volume using the equation that is shown on the right over here. Okay? By multiplying your average number times 25 large squares times 50 to get the total number in the chamber, and then dividing all of that by 0.02 millimeter cubed, you get a concentration of cells in units of cells per millimeter cubed. That's not particularly useful for most calculations in microbiology, when what we want is a concentration that is expressed in number of cells per milliliter. Given that a milliliter is equal to a centimeter cubed, you can use a simple conversion factor to convert your answer to cells per mil. We're going to work an example of this in the next slide. Let's say you count 12 cells in one of these large grids, and then you go on to count a total of 4 more. So your counts are 12, 13, 11, 12, and 12. You take the average cell count, and that's just done by adding these up. All divided by 5. It's a very typical number to use to derive the average for this sort of process. And you get an average of 12 cells per large square. Those were the double-bordered squares that I mentioned in the previous slide. Now you want to determine the number of cells that are in the entire chamber. You know that there are 25 cells in the entire chamber because I told you that. So you take 12 cells per square times 25 squares. Okay, and you're left with 300 cells. That's the number in the chamber. Okay, that still doesn't give us a concentration, right? Like a number of cells per volume. So in order to determine the concentration, you need to factor in the volume of the Petrov-Hauser chamber. So you take the 300 cells and you divide it by that volume, which, as I told you a moment ago, is 0.02 millimeters cubed. And that's going to give me 15,000 cells per millimeter cubed. And as I mentioned a moment ago, that's not a particularly helpful unit. So I want to be able to convert millimeters cubed to centimeter cubed. How do I do that? What's the conversion factor? Well, 1 millimeter cubed equals 1 millimeter times 1 millimeter times 1 millimeter. Agreed? Oops, sorry. So that means that if you know the relationship between a millimeter and a centimeter, which you should if you're familiar with the metric system used by scientists across the world, you know that there are 10 millimeters in every centimeter. So 1 millimeter is the same as 0.1 centimeters. So 0.1 centimeters by 0.1 centimeters by 0.1 centimeters is the same equation, okay? And that's going to equal 0.001 centimeters cubed. Okay, so I can convert now. 15,000 cells per millimeter cubed times the conversion factor, which is 1 millimeter cubed, all divided by 0.001 centimeter cubed. These go away, and I'm left with 15 million cells per centimeter cubed. Okay? And the nice thing about that is that I remember, because I'm familiar with the metric system, that 1 centimeter cube, or cc as it gets called in health fields, equals 1 mil. It's a useful conversion factor. So I'm going to rewrite that in scientific notation and convert it. 1.5 times 10, 1, 2, 3, 4, 5, 6, 7 to the 7 cells per centimeter cubed times 1 centimeter cubed per 1 ml gives me 1.5 times 10 to the 7 cells per mil. That's my concentration. Okay, so that's the concentration that was in the solution that I was measuring. If I had 10 mils in that solution, I could then multiply 1.5 times 10 to the 7 cells per mil times 10 mils, and I would know exactly how many cells I had in the total sample. That's how to use a Petrov-Hauser chamber. Okay, so a lot of times we're trying to count samples in solutions that are not simply pure cultures where you just have a liquid and then cells in it. You might be trying to count the number of samples in a soil sample or a sediment sample or lake water, and so there's debris in there. So a really common method that microbiologists use to deal with that is fluorescence microscopy. So they are still counting cells using the microscope, but they're dyeing those cells with a fluorescent dye that binds DNA or other cell molecules either generally or specifically. It's the same principle as using a Petrov-Hauser chamber. So it's still cell counting, but it can resolve cells from environmental debris by the fact that the cells will fluoresce and the debris should not. So it's very useful in examining real-world samples. There are also forms of fluorescence microscopy methods that can distinguish between live and dead cells, and also specific genera or species. This is a pretty evolved area of microbial ecology. So the last method that I'll mention that is a direct method for counting cells is the flow cytometer. A flow cytometer works only on some sample types. It can only work well on cells that are relatively large, and it does not work well on cells where there is debris present that can be mistaken for cells. Basically, you have a laser light source, you pass your sample through a tube that the laser light source is hitting, and you have some stained cells, so there's some principles of fluorescence microscopy here. The stained cells will reflect fluorescence, emit fluorescence light, and that can be detected by a detector and counted using an algorithm and a computer associated with this device. So microscopy as a measurement tool has some advantages and some disadvantages. The advantages are that it's very rapid. There's no need to wait for anything to grow. The disadvantages are that it counts microbes that, and another advantage is that you can count microbes without culturing them, which means even if you don't know how to culture them, you're able to count them. Disadvantages are that you can't distinguish live from dead cells unless you're using certain fluorescent stains. It's also difficult to use with particularly tiny cells because you're confined to the 1000x magnification of a typical microscope. And it doesn't work well for low density cell samples unless you filter them first and then use fluorescence microscopy. So there are ways around that disadvantage. It can also be really hard to tell what a cell is versus garbage or junk in the sample, for example, soil particles. So let's talk about indirect methods for counting cell numbers. So one example of that is what is known as the viable count method. So these are methods that rely on growth. Okay, so viable counts require the ability to grow microbes in the lab. They can be done under aerobic or anaerobic, which means oxygen-free conditions, at a variety of temperatures and using a variety of media. Generally, the choice in the culture conditions depends on what the scientist most wants to measure. Given the metabolic diversity found within most microbial communities, it is simply impossible to have one set of culture conditions that will allow you to grow all of the microbes in the sample. Nonetheless, this method is really useful for many applications, such as enumerating the number of fecal coliform bacteria in a sample of milk or water or other materials. The top portion of this figure shows the spread plate method, where a liquid sample is pipetted onto an agar plate, spread evenly around, and then allowed to grow. Individual colonies are then counted. A colony is the visible growth derived from what is presumed to be one cell, but typically referred to as one colony forming unit, or CFU. Colonies are visible to the eye because there are so many microbes growing within each one. The lower portion of the figure shows a pour plate method, where a liquid sample is pipetted into an agar plate, and then you add the solubilized liquid agar, so it's still warm, into the plate. You swirl it around to mix and let it set and grow. When you use this method, you'll have colonies on the surface of the plate and also within the plate. These are called subsurface colonies that have to be counted. The pour plate technique is a little more traditional, and it does work well for organisms that use less oxygen, so it may have some advantages in that regard. To make this method quantitative, you have to be able to actually count all the colonies on the plate and then relate the colony count back to the original sample. Let's consider how this is done. So here we have a sample of something where one wants to know how many target microbes are within the sample. So you've got something and you want to count the microbes in it. What do? The first step is to make a serial dilution. This is accomplished by adding 1 ml of the target sample to the first tube. It's labeled 10 to the minus 1. That's effectively a 1 in a 10 dilution. So you've added your sample to that. And then you mix it really, really well. Next, you take 1 ml of that and you put it in the next tube. Note that each of these tubes has 9 ml in it. So every time you add a ml to a 9 ml volume, then you get a total of 10 ml and you effectively achieve a 1 in 10 dilution. So the next tube represents a 1 in 100 or 10 to the minus 2 dilution of the original sample. You mix that well, you take a ml from that, you put it in the next one. Mix it well, take a ml of that, put it in the next one, and so on, and so on, and so on. So there are a total of 6 dilutions shown here, ranging from a 1 in 10 dilution to a 1 in 1 million dilution, or 10 to the minus 6. Next, what you do, and this is a pour plate method that is illustrated here, next what you do is you plate 1 ml of each of those samples. So you take a ml of that and you put it into some agar and you swirl it around and you let it grow. Okay, so you do that for each of the dilution tubes all the way across. Okay, and you have to wait. So there's some time in here. The amount of time needed is whatever it takes to incubate long enough to grow cells. So it depends what you're studying that time. Okay. Then you look at the plates, and what you'll see, you're looking for plates that have between 30 to 300 colony forming units. Okay, remember, a colony is the visible growth from what is presumed to be one cell. So these have too much on it, they're called too numerous to count. Right? That's how you would score that data. But here we've got countable colonies. So we've got 159, we've got 17, we've got 2, and then we've got none. In this sample, we've basically diluted to extinction. And that's good because we know then that our sterile technique is good, that we're not accidentally introducing microbes from the environment. This is the only one that's between 30 and 300, this plate that has 159 on it. So that's the plate that we're going to use. We use that count, we multiply it by the dilution factor, which was 10 to the 3 or 1,000, and what we get then is the number of colony forming units per milliliter of the original sample. Okay, it's 1.59 times 10 to the 5. Now something that's a little weird in microbiology, but you'll just have to trust me on this because many years of experiments have proven it to be true, is that when you're applying this concept to the spread plate methodology, where you take a sample and you just spread it onto the plate rather than mixing it into other agar and pouring it around and then letting it grow. So in this case you would get colonies only forming on the surface of the sample. It in effect adds another dilution factor. Okay? So if we were doing that, despite the fact that this dilution represents a 1 in 10 dilution, the act of spread plating is another 10-fold dilution. So in effect, the dilution that we would be seeing on this plate would be 10 to the minus 2, and this plate it would be 10 to the minus 3, and so on down here. This would be 10 to the minus 7. And when we were doing the calculations then, the dilution factor wouldn't be 10 to the 3, it would be 10 to the 4. Okay, so that's just something to remember for future use. Alright, so let's solve this. You want to know how many bacteria are in a sample of milk. You are using the viable plate count method, and the agar you will use is milk agar. You are using the pour plate method. Okay, you prepare a dilution series of 6 tubes. The first tube is a 1 in 10 dilution, and the sixth is a 1 in 1 million, just like the previous example. The table below shows your data. The dark blue is the dilution tube used, and the light blue is the number of colonies counted. How many colony forming units were in the sample? Well, we'll look at it and we'll see which one is between 30 and 300. It's this one, okay? So that's the count that we're going to use. What's its dilution factor? Well, it's 1 in 10,000. So effectively what we do then is we multiply the count by 10,000. And we get 1, 2, 1, 4 zeros, so that's 1,210,000 colony forming units per ml, okay? Or 1.21 times 10 to the 6 CFU per ml. When you're trying to use viable plate counts to assess the number of cells that are in a sample where that cell number is probably really low, where the concentration is really low, then a common method is to use what's called filtration. Okay? So you collect a water sample and you pass it through a filter. And the filter will usually be 0.22 micrometers in diameter. Or so. It's enough to capture most known bacterial and archaeal cells. You filter the sample, you grow the filter on an appropriate media, and appropriate means that it depends what you want. If you want to grow fecal coliform bacteria, you grow it on a media that's optimized for fecal coliform bacteria. If you want to grow anaerobes that are able to use nitrate as a terminal electron acceptor, then you grow it on media that has nitrate present, and you put them in no oxygen conditions. It just depends. You grow them for a while, you watch for the formation of colonies, you count the colonies, and then you take that count and you divide the count by the volume that you filtered in order to obtain a concentration of cells. So one thing that's really interesting that was first discovered back in the 1950s by Janosch, a very famous microbiologist at Woods Hole in nearby Massachusetts, is something called the Great Plate Count Anomaly. And what the Great Plate Count Anomaly means is that when you compare cell counts obtained using microscopy and cell counts obtained using viable plate counts, the numbers don't match. And in fact, what we see is that the numbers for viable plate counts are much, much lower than those obtained using microscopy. So what does this mean? It means that plate counts only reveal a fraction of what is there. And if you think a little bit about the metabolic diversity of microbes found out in the world, the reason that we have the Great Plate Count Anomaly is that you can't have one single culture medium that will allow the growth of all the different kinds of microbes present. And in fact, we're still really discovering new metabolic abilities in microbes, so we're not yet knowledgeable about all of the kinds of microbial media that we could be making so that we could potentially grow everything within the sample. And so this is something that microbiologists have developed methods to get around. We have a lot of other ways of assessing microbial communities, for example, examining the DNA fingerprints of different species present in communities. And then we're also getting much more creative about types of culture media that we can make based on some of that genetic data. But it's important to be aware of the Great Plate Count Anomaly. It's an important issue in microbiology, past and present. So let's talk briefly about indirect counting methods. So these are methods that do not rely on growth per se, or do not rely on directly visualizing cells or colonies per se. So methods that are indirect are turbidimetric methods, assessing cell mass, and assessing chemical levels. Cell mass would mean just weighing cultures, taking the dry weight of them over periods of time and using that to show population growth. Chemical levels might be measuring the level of ATP, of DNA, or of protein. Quantitative PCR, which is a technique used to evaluate copy numbers of particular genes, is an example of a chemically based indirect counting method, as is ATP quantification. Let's think a little bit about turbidimetric methods. So in turbidimetric methods, we use a spectrophotometer. We take light and we shine it through a prism and a filter system, and that allows light of particular wavelengths to pass through a sample. So that's light. The light that's coming through here is of a particular wavelength. And the wavelengths most commonly used for this purposes are 480, 540, 600, or 660 nanometers. Nanometers is the unit used for measuring the wavelengths of light. What'll happen is some of the light will hit cells, okay? And when it hits cells, it will scatter, all right? The unscattered light will make it through to the detector, and the photocell or detector will measure the unscattered light, okay? That's what the spectrophotometer is doing. When we're using spectrophotometers in this way, we usually use the term optical density rather than absorbance. And this is simply to acknowledge the fact that the cells are not actually absorbing light the way that a chemical would, but rather scattering the light so that the incident light that penetrates into the detector has not been scattered. So it's a little bit of a small differences in terminology to acknowledge the physical reality of the system. What's great about these methods is they're really quick and really easy. So you can get data really quickly. And you can use that data and you can plot a growth curve, which we've discussed previously, and you can calculate generation time quite simply from the growth curve. So you can get a lot of really useful information using these methods. What you don't get is you cannot actually correlate cell number to that data unless you use another method in conjunction with it and develop a standard curve. With that, I will conclude this discussion of growth methods, of our methods for assessing growth.